I'm Matteo Blankensteiner, a postdoc fellow of ETH, and you are wondering now why I have a blue lab coat. The truth is that in the laboratory of Professor Punk at the Bioprocess laboratory, we are working on developing a completely new synthetic route for a clean indigo. Indigo is the main ingredient for the staining of denim, what is our blue jeans. Indigo is a natural product, is isolated from plants and is being used for centuries from those sources. However, for coping the high demand of the last century, chemists develop synthetic ways which are more efficient and can produce a very large quantity of such dye. Unfortunately, such synthetic routes have some pitfalls. They produce toxic compounds which are harmful for the health of human beings and for the environment, but also they rely on carbon source from fossil fuel. In the last years, there's been a rise in interest in sustainable production of chemicals. Also in the textile industry, there has been this rise in eco-conscious synthesis of dyes. And it is here where we place our research. We want to find a way to produce indigo in a very clean and sustainable way. It took 3 years to find the right solution. We took the genetic code used by plants, we installed it in a bacterial cell, E. coli, we synchronized it with the metabolism of the cell, and we obtained a cell that was able to produce indigo at a significant amount. You can see here on those bioreactor, we have our indigo producing strain, which is growing, is fed with glucose, which is a sustainable carbon source, and in about 2 days, it will generate a titer of indigo, which is comparable with the industry demand. As you might see, those is a laboratory scale, and is clearly not the amount that the industry wants. They look for very large quantities. So we were curious to see if our strain could perform well also at larger volumes. We went to a pilot plant of 0.5 meter cube. We ran our experiment. We put our bacterial producing strain in a larger fermenter. We ran the same experiment. We fed the glucose, and we were happy to see that the strain performed equally well as the laboratory scale. We could produce about 5 kilos of indigo. We could isolate it equally well, and we obtain also completely clean, 100% clean indigo in a scale which we think have never been done before using bacteria.